Hello, everyone, and welcome to today's keynote webinar titled Modeling the Holistic Journey of Solid Tumor Cell Therapy with Organ Chips. This webinar is a part of the third annual Immuno-oncology virtual event series. I am Sydney McNeil, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and sponsored by Emulate Bio. For more information about Emulate Bio, please go to www.emulatebio.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many questions as we have time for at the end of the presentation. If you happen to have any trouble seeing or hearing the presentation, please click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now let's get started. I now present today's speaker, Anita Mehta, Principal Scientist at Emulate. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Anita, you may now begin your presentation.
Hello, everyone. Thank you so much for joining us today. Before we talk about CAR T cell therapy, I want to give you a quick introduction on Emulate. At Emulate, we develop organ-on-chip technology for use in disease modeling and drug discovery. So let's take a look at the organ chip. Inside we have two chambers, the top channel and the bottom channel. The top channel, you can see your epithelial cells. The bottom channel, you can see your endothelial cells. Both of these channels are separated by a thin porous membrane, which allows cell-to-cell interaction. And in the vacuum, the stretch can be applied to mimic the actual stretching or breathing pattern. On the both of the sides of the membrane, you can coat them with the tissue matched ECM, and vacuum. Then within each channel, you can flow distinct media or establish an air-liquid interface and collect effluent for a downstream analysis. So now, at Emulate, we haven't just designed the chip, we have created a complete user-friendly solution, which we call human emulation system that combines the consumable, the hardware, the software, all the researchers basically can create a human relevant and translatable organ chip models, regardless of their experience with advanced cell culture techniques. Now, let's look at the consumables. The consumable include the chip, which we just saw, as well as the pod, which is the portable media reservoir for the chip. It basically allows the user to easily add or change media, control any compound dosing, and sample effluent for further downstream analysis. Now, let's have a look at the hardware. The hardware provides a robust and seamless organ chip interface and can house up to 12 organ chips. And it can allow you for the user can basically do their own flow rates and cyclic stretch as needed. And you can see here the Zoes that sits inside the incubator and can house up to 12 chips. And on this is called the orb. It sits outside of the incubator and is used for gas exchange. And this can basically serve the 4 Zoes. Now, we also have software developed in-house that allows the users to basically do data analysis or a high throughput image analysis. We also provide calculators to support any of the study design, and also effluent collection or analysis for understanding the mechanisms of action. Now, but this is not. But this is just. This is not all. We have developed a number of organ models to study toxicology, disease modeling, and efficacy applications. So this basically includes models of a small and large intestine, the proximal tubule kidney chip, the liver chip, the colon intestine chip. All of these models has been well characterized, has been very much validated, reproduced, and are pre-qualified to understand biology. But that's just a fraction of what's possible with the Emulate system, with the human emulation system that Emulate had developed. There are over 100 publications using the Emulate technology, and some of the most exciting use cases come from our user community. Now, let's take a look at how it can be applied to study CAR T cell therapy for solid tumors. So before diving into the detail of the workflow, let's first see what CAR T cell therapy is. So CAR T cells are basically engineered chimeric antigen receptors. These basically would recognize the tumor cells or the antigen expressed on the tumor cells and specifically kills those tumor cells. So these CAR T cells have revolutionized the way we have thought about the cancer treatment in the past. That led to 6 FDA approval for the liquid cancers. As you can imagine, there's only 10% of cases that represents liquid cancer. However, majority of these cases are solid tumors and accounts more than 90% for solid tumors. The way CAR T cell therapy works is usually the T cells are isolated from a patient and then modified by using genetic engineering, either by lentiviral or mRNA-based therapies. And then it's infused back into the patient, where then in the bloodstream, those targeted T cells basically look at the cancer cells and kills them. However, unfortunately, the efforts to adapt CAR T cell therapy to solid tumors has proven challenging for a number of reasons, which we will talk in a little bit. So, there are around 8.5 million annual cell death from solid tumors, which is way higher than we think for liquid tumors. And there are around 400 CAR T cell clinical trials for solid tumors, and none of them have been approved by FDA to study CAR T cell therapies for solid tumors. Well, why is that the case? Now, let's look at some of the challenges that are basically focused on solid tumors. As we know that liquid tumors have fewer antigen and they are more homogeneous compared to the solid tumors. Whereas there is one of the major factors is antigen heterogeneity in solid tumors. They basically express a myriad of antigens on their surface, which makes it challenging to target them. On the other hand, we know that liquid tumors don't have an immune suppressive tumor microenvironment. However, the solid tumors, there are multiple factors that limit the efficacy of even if the CAR T cells are there. The third factor is the CAR T cell exhaustion, which means that even though the CAR T cells get infiltrated, it might not be capable of killing those tumor cells. So But really, none of these three factors matters unless the CAR T cell actually reaches the solid tumor, which is why today we want to focus. Today we want to focus on the challenges of CAR T cell trafficking and infiltration. So for blood cancer, CAR T cells are administered directly into the bloodstream and where the cancer is, and they can recognize it and kill it. For solid tumor, it's a different journey. They have to basically come out of the bloodstream and then get migrated into the tumor, and that's the CAR T cell trafficking is critical. However, a very rate limiting step that is often overlooked. So conventionally, the 2D models can basically give the killing of the tumor cells at the end of the journey. It basically lacks the entire journey, which we just went over, which is needed for the cell to exit from the bloodstream and enter the tumor. So the solution to better understanding of CAR T cell therapy is our flexible organ chip workflow to evaluate CAR T cell recruitment and killing, which allows you to model the complexity of human immune responses to recreate this holistic CAR T cell journey. Now you will see a video of how this model works.
Specific microvascular cells. Inside this lung cancer model, CAR T cells are flowed through the vascular channel. In response to the localized inflammatory conditions, CAR T cells attach to the endothelium and migrate across the vasculature to the target cancer cells. Here, the chimeric antigen receptor enables the T cells to recognize, bind to, and kill the cancer cells.
Okay, so as you saw in this animated video, this basically shows the workflow in a video format. So what we do is we flow the CAR T cells in the vasculature channel under high flow rates in response to the localized inflammatory conditions, which I will go in a little bit detail in later of the presentation. The CAR T cells then gets attached to the endothelium. They migrate across the vasculature to the target cancer cells, where the CAR Ts then recognizes the antigen that is present on the tumor cells, binds there, and kills the cancer cell. Now, let's look at the process in a little bit detail. I know the video was short. So what we do in reality is like we seed the epithelial channel, the top channel with the epithelial tumor cells, the bottom channel with the vascular, the tissue matched endothelial cells, which we call vascular channel. And then we basically add the priming stimulus. In the bottom channel, we prime the endothelial cells with TNF-alpha, and the tissue-relevant chemokines are then added on the top channel at the same time. So CAR T cells are flown, and the TNF-alpha primed endothelial cells help in the adhesion molecules of the CAR Ts. Then the CAR T cells gets attached to those adhesion molecules and then migrates over the membrane to the top channel, where they have their final destination, the tumor cells, and they basically recognize those antigens on the tumor cells and then kills them specifically. Okay, to capture the holistic journey of CAR T cells, we have developed a flexible workflow that can be adapted by the user to evaluate any target cells of their interest with any of the CAR Ts of their interest, and can also be adapted to the co-therapies of interest of the user. So now to develop this workflow, we have used non-small cell lung cancer because it's one of the leading causes of cell death. One of the leading causes of death related to cancer, solid tumor. We used A549 that represent a lot of the antigens that can be targetable and clinically targetable antigens are expressed by the A549 cell lines. We used CD19, which is not present on the A549 cells. It's specifically expressed by the hematological cells like B cells. We used a second generation CAR that we acquired from Promab that has HER2 as well as CD19 CAR. These CAR T cells were administered via tissue-specific vasculature that we went over few slides back. These cells are commercially available and can be used in the same workflow. And then also the co-therapeutic, one of the challenges in cell therapy space is the basically having the efficacy as a monotherapy. So we basically provide a proof of concept by using IL-2 and combining that with our HER2 CARs. So with this flexible, the users can basically use their own tumor cell line of interest, the CAR T of interest. What they will be able to achieve is basically a tumor vasculature interface, also co-culture of their tumor cell line with tissue-matched endothelium. They would be able to achieve the CAR T cell recruitment and killing, providing the holistic journey of CAR. They would be able to analyze the endpoint looking at the effluent, the confocal imaging, immune cell extraction, and phenotyping. So let's look at the results now. We used A549, as I mentioned earlier. A549 cells express HER2 antigen. They lack CD19 antigen. And shown here is the mean fluorescence intensity. And also what is interesting is that the cytokine chemokine priming did not alter the antigen expression of either HER2 or CD19, which makes it ideal tumor cell line to test these CARs. Now, here is the Z-stack of a 3D confocal imaging micrograph, and these are the segmented images show that you can basically look at the journey of your CAR T cells, how many total cells got attached to the endothelium, inside the membrane, and on top of the channel where the tumor cells exist. Now, let's look at the data. You can basically create these stacked bar graphs. So shown here in the light blue is all the CAR Ts that got attached in the bottom channel. The lighter blue is the CAR Ts that are inside the membrane. And then on top of the channel are the ones that's shown here with the darker blue. What you can appreciate is that with the priming stimulus, you see more of the recruitment of the HER2 CAR T when they see the HER2 antigen on the tumor cells that are present on the top channel. We do see CD19 migrating to the top channel, but to a smaller extent compared to the HER2 CAR T cells. So now let's look at the killing, which basically provides the efficacy or efficiency of the CAR T. So what you can appreciate here is that even though these CAR T cells are labeled with the cell tracker dye, the red signal represents the CAR Ts that are on the top channel. You can see that both HER2 and CAR T and CD19 CAR Ts do migrate on the top channel. However, what is is really striking is that you don't see that killing, which is shown here by caspase 3 and 7, and is labeled with the green color. So you can see that HER2 CAR, when it sees the antigen, it specifically kills the tumor cells, and that is absent in CD19, which basically tells you the specificity or antigen-specific killing of these tumor cells, which is not seen with the CD19 CAR T cells. So what happens upon the engagement of these T cells with the tumor cells is that it creates a cytotoxic synapse, which then leads to the degranulation of granzyme B, perforin, interferon gamma, and interleukin 2. So in our model, we also wanted to investigate if we are able to see the T cell specific degranulation and then subsequent tumor cell killing. In order to do that, we developed MSD or effluent-based assay, where you can look at the degranulation of certain cytokines like perforin, granzyme B, interferon gamma, and IL-2, and the trends look similar to what we have seen with the caspase. So the reason of doing this is that you can basically look into the mechanism of action and see that it's the killing of the tumor cell is basically specifically due to the release or degranulation of these cytokines. One other opportunity that the user can utilize is looking at the time-dependent and kinetics of the release of cytokines or degranulation of these cytokines. So what we did here is we collected the effluent at different time points. Shown here is 24-hour, 48-hour for the HER2 CAR, which is the plus means primed with the chemokine cytokines, and the minus means it's not primed. So as you can appreciate here, over the time, this would indicate an early stage killing. The 48 hours would indicate the late stage killing. So you can basically look at the kinetics of the killing in a time-dependent manner, which can open a whole new avenue for looking into the mechanisms of action of these different CAR Ts and combining different CARs and looking at the designs of different CARs, how that can affect the degranulation process. This workflow also provides users with immune cell extraction. So here on the left-hand side, you can see that these are PD-1 expression on the cells that got recruited to the tumor site. The reason you see more on the HER2, because they have basically encountered the antigen on the tumor cells, killed it, and then got exhausted. So as you know, that PD-1 can be either used as an activation marker or an exhaustion marker. That is also coupled with the soluble PD-1 that is shown here by effluent analysis and using by MSD. So this method can basically give you the flexibility of looking at the terminal endpoint, which is the extraction, and also by looking at the effluent, and then really correlate and understand the mechanisms of action for your CAR. So in terms of monotherapy in cell therapy, we know that there are challenges of the durability of the CAR. So by using IL-2, which is very well studied cytokine that can give a boosting signal to the CAR and make it more cytotoxic. So we wanted to combine that with our monotherapy CAR, and we have done a proof of concept by adding IL-2 with our HER2 CAR and seeing what outcome would we get from this. So what you can see on the left-hand side, by adding IL-2, you see a little bit more increase in the total T cell migration to the epithelium compared to the monotherapy HER2, which is absent in CD19 cases. Although it's not significant, what you can appreciate is that the killing response is robust. So even though there is no significant enhancement in the recruitment, but it basically adds in the or boosts the fitness of those T cells to kill the tumor cells. So if you would look at just at the recruitment, you will miss the piece of the efficacy of the CAR T. So you basically have to look at the holistic journey. That's why I think it's important to look at how these cells are getting recruited and then killing them in an antigen-specific manner. And that is coupled with degranulation of the cytokines, looking at perforin, granzyme B, and interferon gamma. You see there is robust increase with the degranulation when we combine them together. So in nutshell, what this CAR T workflow provides, it provides a validated and flexible CAR T workflow in a unified assay. You can basically look at the CAR T vasculature attachment, recruitment, and eventually killing, which is the efficacy of the CAR. In antigen-specific killing and degranulation, we also provide proof of concept co-therapeutic efficacy that can be modulated based on the user need. We can also provide with the CAR T cell extraction and immunophenotyping analysis, which is, I think, is one of the key factors for cell therapies. Now, what can we leverage this workflow? You can it basically opens the whole entire new avenue where you can rank order the efficacy of the CAR T cells and add in different co-therapeutic agent and look at the mechanisms of action, expand this model to include like more complex, cell types, immune suppressive, tumor microenvironment to really model and recapitulate what happens in the human scenario. And also, this can be expanded to assess therapeutic safety, like on target of tumor that we face currently in a cell therapy space. So, with this, I'm happy to take any questions, and thank you.
All right, thank you, Anita, for your very informative presentation. We will now start the Q&A portion of the webinar. If you happen to have any questions you would like to ask, please go ahead and do so now. Just submit a question in the Ask a Question box located at the bottom of your screen. All right, let's go ahead and get started. Okay, the first question that we have here is, have you tried using co-therapeutics other than IL-2?
That's a great question. We actually have not tried that yet. Yeah.
All right, perfect. We have another here. It says, how did you choose which chemokines to prime the epithelium with?
So Emulate did a deep dive into literature, and we came up with the most clinically relevant chemokines that are required in the setting of non-small cell lung cancer.
All right, thank you. The next question that we have here says, what other immunology applications are possible with organ chips? What are the ranges of diseases that you can model?
Yeah, that's a great question. So this is a highly adaptable workflow, and you can model basically, the both type of immune components, including the tissue resident. We have done that in the past on the liver model, where we used Kupffer cells that are the tissue resident macrophages. You could also do the circulating immune cells, which we also launched the immune cell recruitment application some times ago that basically uses IBD model, and then the CAR T is also with where you can use the circulating immune cells.
All right, perfect. We have another here. It says, have you explored the use of other types of CAR T? For instance, have you tried targeting other antigens expressed by the cancer cells being utilized in this application?
So that's the beauty of this workflow. It's adaptable. We have not tried internally any other targets other than the CD19, which is a negative CAR that we used, and as it's not expressed by the solid tumor. We did HER2, which is targeted towards the solid tumors. We have not done any other CAR-Ts. However, we do describe in the detailed protocol how to adapt this workflow for the based on the user needs, and we will be happy to help in like setting up and we also have a troubleshooting guide section in the protocol, which is also something that user can go through with that, and we can definitely help setting that up.
All right, amazing. Thank you. It looks like we have a few more that have come in here. The next is why is a high flow rate needed for CAR T cell administration?
Yeah, so if you think about in the in vivo setting, there is, the recruitment of the immune cells is highly selective process. So you need high pressure or high shear forces to kind of, have the selective recruitment or migration. And the high flow rate was basically dependent on the in vivo situation where we see where the pressure is 2 dyne per centimeter square, but the flow rate that we are using in this workflow is equivalent to that, which is 0.3 dyne per centimeter square.
Okay, perfect. How would I adapt this workflow to a different CAR T or cancer cell type?
Yeah, that's a great question. So we do have a very thorough description in our protocol, which basically walks the user step by step, where what you need to do is like look at the expression of the target antigen, then like look into the CAR T of interest. Then we also basically walk you through the plate studies and what are needed in terms of like the making them more priming stimulus, doing a literature search for chemokine cytokines, and the chip studies as well. So, and we would be happy to guide the users through if someone is interested to adapt this workflow.
Perfect. Yeah, that's great to know. Okay, it looks like we have two more questions here. So we will go ahead and wrap up with these last two. Second to last question here is, can you extract the recruited immune cells for flow cytometry analysis?
That's a great question. Yes, we indeed did that with our workflow. The user are able to extract the immune cells. We do the enzymatic digestion for extracting the immune cells, and you can definitely look into the markers of interest.
All right, perfect. So we will go ahead and wrap up with this final question here. Is this workflow compatible with patient-derived organoids, PDO, or a model of the tumor microenvironment?
That's a great question. We have not tested that in-house. However, this workflow is highly adaptable and can be done with the patient-derived organoids. We do have chip A1, which provides a larger platform where, and you can use like different type of cells to kind of mimic the tumor microenvironment. So definitely this workflow can be adapted to model the tumor microenvironment.
All right, perfect. Thank you, Anita. Do you happen to have any final comments for our audience today?
Yeah, I would just reiterate that this workflow is great, and as we know that the CAR Ts are revolutionizing the treatment of solid tumors, and this would basically really help the users to kind of rank order their cars and use this workflow to really understand how a CAR T can be used for tumors, for solid tumors rather than just liquid tumors.
All right, wonderful. Well, thank you again, Anita, for your time today and for your very important research. Before we go, I would like to thank the audience for joining us today and for their very interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until April 30th, 2025. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.